Okay, coming back to the discussion on that biocompatibility and host response. The previous discussion has been summarized in this slide to some extent. So, one has to start with this lab scale material synthesis or fabrication of the simple shaped samples. Now, simple shaped samples can be either cylindrical shaped or can be rectangular shaped and so on, which may not be the ultimate shape of an implant which will be put it inside the human patient. So, let me reiterate this statement. What I am saying is that in the laboratory, whatever biomaterials or scaffold we synthesize, normally for the in vitro testing purpose or for physical property measurements purpose, these samples are of simple geometric shape, which in no way can be can have any similarity in terms of the size or shape of the implants that are to be placed in human patients. So, however, that why we start with the simple shaped material, because simple shaped material is required for standard material testing, like whatever strength property you are testing or hardness property or fracture toughness property or even microstructural phase assemblage study. You need very simple shaped material, and so that you can establish what I called is a proof of concept. Proof of concept essentially means, like first of all one has to know that whether a material is having appropriate compatibility at cell and tissue level. So, now to prove that point you can do this testing with a simple shaped material. After that, you need to do is that in vitro compatibility test, cytocompatibility analysis we using various biological assays. Now, these biological assays are actually kind of scientific experiments where one can use the elements of a biological system, be it protein, cells, blood sample or bacteria, with an endpoint objective to determine its functionality change or its survivability in contact with a biomaterial or artificial material or scaffold. And this is to be done with the glasswares or test tubes or petri dishes, as I have told you before, and this is done in a typical lab scale experiments. Okay. Next level, once you select some of the material, so you select a material which from a group of material which will have the best combination of both physical property and in vitro cytocompatibility property. You select that material and then go to the next stage of research, that is called in vivo biocompatibility assessment, and this is also known as I said before, preclinical study. Now, the rationale of doing this preclinical study I have mentioned you before. One is to start with a small animal model, then go to medium and large animal models to validate the biocompatibility results which are obtained with the small animal model. So, here that animal ethical committee approval is a must, and each institution wherever in this world wants to carry out this animal testing must have the institutional animal ethical committee which has members from different disciplines as well as socially conscious person as well as person who is remotely linked to this study. So, that particular committee is given the assignment to approve any animal study, and that with that approval one can conduct this preclinical study in the animal model. And this approval becomes much more complicated or even much more strict if one goes from small to medium to large animal model. For example, if one is to carry out the experiments in a monkey model, the approval is most difficult, but if one has to carry out the experiments in a rat or mouse model, the approval can be little easier, provided the study design is appropriately framed or study design is appropriately rationalized within the framework of the guidelines of the standard bodies like ISO guidelines or British standards and so on, or FDA approved protocol one has to use. So, once the preclinical study is done, animals survive, bone healing has been established, or there is no undesired tissue response because of the implantation of a synthetic material that you are investigating. The next level is that one can do this prototype development of the biomedical devices, and this prototype development has to be, and after this prototype one has to do further testing, and what I called this testing at device level. Now, just to give you two examples in the context of orthopedic or bone tissue regeneration applications or bone tissue engineering applications, one is a THR and another one is TKR. THR stands for total hip joint replacement, TKR stands for total knee replacement device. Now, in both the cases THR and TKR, the final device has a much more complex and complicated shape and size than one you start with a simple shaped test sample at the laboratory scale. That is the step number 1 in your entire biomaterials research. So, once this femoral ball head or acetabular socket assembly is fabricated, they are being assembled and so on, one has to carry out device level testing using joint simulator, and there one can simulate that when a person walks, what is the stress level, what is the stress cycle, then that a device is expected to experience. So, that stress cycle one has to simulate within the joint simulator, and there are two joint simulators which are relevant. One is that hip simulator that is relevant for total hip replacement, and another one is the knee simulator that is relevant for total knee replacement devices. So, once a large number of fatigue cycles are conducted using this hip simulator and the materials survive, materials do not fracture, they do not wear to a significant extent, and their wear debris particles are taken and their toxicity has their non toxicity at cell or tissue level has been established, then only all these preclinical test results is further transferred to the next level of study, that is called clinical trials. As I said, clinical trials essentially is extremely difficult because of the requirement of the strict regulatory approval. Now, clinical trials, again one has to write a proposal, and that proposal has to be approved not only by the institutional ethical committee, but also national level ethical committee. In India, we have the DCGI, that is Drug Control General of India. So, DCGI kind of approves all the clinical trials that are carried out on Indian human patients, and similarly every country own national level approval committee for clinical trials, because clinical trials often come to the newspaper, because if the clinical trials are conducted in not an appropriate manner or not a clinically acceptable manner, then it may cause kind of very undesired results, and it can lead to the death of the human volunteers, which is not at all desired, and often it comes to the newspaper this the wrong clinical trials being conducted in different parts of the world. Now, once the clinical trials are done, then the device is ready, the technology is can be transferred to the industry, and therefore, that commercialization is possible. Okay. So, before that host response, before one can assess the host response, one has to do in vitro testing, and in vivo experiments one can assess the host response, right. So, this slide is kind of important in a sense that this slide essentially tells you that what is the difference between in vitro and in vivo experiments. To repeat, in vitro means lab scale simulated experiments. However, it is a poor representation of a physiological condition. There is no inflammation that you can study here, there is no immune response. Largely in vitro experiments are conducted with single cell type. Either you culture osteoblast cells or you culture fibroblast cells, but there is a new concept or recent trend people try to use that is called co-culture. Co-culture means two different cell types are cultured together. Okay. And so, this is, but this is still not being regularly used in different biomaterials laboratory. The other things that you cannot do or you cannot assess in the in vitro experiments is the tissue remodeling, simply because you are handling with the biological cells only. In contrast, in vivo experiments, that is the animal experiments, are better approximation to human environment. Let me spend some time on this particular first point, that what is better approximation of the human environment. Essentially, human physiological environment or human bone structure or bone composition or bone characteristics. Now, the way that human osseous structure can be defined depending on the anatomical location, natural bone has slightly different variation in terms of the composition as well as the properties and certain characteristics. Now, the several animal models are used, but out of that 3 animal models I must mention which closely mimic that human bone structure. One is called dog, second one is called sheep, and third one is called pig. Now, dog, sheep model and pig model, these are all, as you see that these are all large animal models. Now, dog model has a bone composition and bone structure, it's and bone and animal weight and all that is kind of coming close to that of that the human model, or it can be used as a representative human model. But here again bone microstructure and bone remodeling of the dog structure is quite different from that of the human system. Pig again is a good model because pig osseous structure or pig bone structure and remodeling is quite good, but pigs, the problem is that their bone size or that femoral size or tibial size is rather small. So, therefore, experimentation or accessibility to that bone structure while conducting pig model in an operation theatre is quite difficult. Also difficult is how you can manage the pig models during experiments. So, these are the real bottlenecks in using pig models. The third one is a sheep model. Sheep model is very good because sheep model has the bone structure is and microstructure as well as bone remodeling is very close to that of that human system, and essentially many people they use that sheep model. And sheep model one can use very large segmental defects, and if one goes to literature then you can find that typically in the sheep model people have used large segmental bone defect, and these defects can have size somewhere between 10 to 48 millimeter. So, this is a large size. So, suppose you have we have a large irregular bone structure you have, and a large bone structure, suppose you cut this part of the bone, then you have an cylindrical implant. This implant you can insert it at this bone defect, and this there is something called critical size defect. So, the definition of the critical size defect is that if you cut a large piece of the bone structure and try to replace it with the implant size, the size of this implants, the length of the implant, the L should be somewhere between 2 to 3 times the diameter D of the host bone, of the diaphysis of the host bone. So, if you maintain this kind of dimension, then it is called critical size defects. Now, one of the challenges in the orthopedic research is to establish that whether these critical size defects can be healed using synthetic biomaterials or using artificial biomaterial. So, I repeat, so this is your natural bone structure. So, if it is a large long bone structure like femur or femur structure, now you are cutting this small segment of the material, a small segment of the natural bone, and in this gap you are putting your implant material, which I am now shading. Now, this implant material, the length of this implant material can be 2 to 3 times that diameter of the host bone structure where it is being cut, then you can call it as a critical size defects, or this kind of defects are also known as segmental defect. And the segmental defect model in general is kind of a little different from the cylindrical defect model. Now, cylindrical defect model, what you have? You have a femur of, let us say, rabbit. In this femur you are drilling some holes, and this typically the diameter of this hole is around 2 millimeter. The length of this implant, which you can put it inside this put it inside here, it can be somewhere between 5 to 6 millimeter. So, that aspect ratio is around 3. So, the length of the implant which can go and get inserted into this defect, cylindrical defect, is around 5 to 6 millimeter, with the diameter of the implant is 2 millimeter. And then you see that around this cylindrical defect model, how this bone regeneration takes place, and how long does it take to heal this kind of bone defects. So, that is the whole gamut or aspect of this host response in this particular defect model. Okay. So, I hope I have kind of explained to you is little bit more details on how you can choose the animal model. So, selection of the animal models is very important, and there one has to find out that what is the typical bone structure of that material, of that animal, and how this bone structure composition or the microstructure is close to that of that human natural bone. Second one is the demanding protocol. So, whatever protocol that you want to use, that should be acceptable to the society also. This is second step prior to the clinical use, very important, and the interactions among different cell types, proteins and biological molecules and extracellular matrix, these can be investigated together in any animal experiment. So, this is by far, I would say, largest scientific advantage in terms of assessing the what I mentioned earlier, that biological acceptance or biological system acceptance to a synthetic material. So, once this is sufficiently being assessed through in vivo experiments, one can finally do the medium or larger size animal experiments to validate what results you have got at the small animal model. Finally, the hormonal effects also, hormonal factors also can be assessed in this in vivo experiments. Let me also summarize some of the earlier discussion in this slide, particularly in the context of the in vitro cytocompatibility. So, this assessment of cell adhesion, growth, proliferation should be done using targeted application specific cell line at different points in time points in culture. For example, you want to do the time in culture, let us say 2 weeks or 4 weeks or 6 weeks, that is the typical time in culture, and then you see that how cell numbers is increasing. So, if it is increasing in a linear manner, then that means cells are able to grow or multiply or divide while being attached to the biomaterial substrate. And how to select that what is the different timelines that you need to be using, that depends on the cell doubling time. So, typical human cell lines has a doubling time of 12 to 24 hours, a little longer. Then depending on the cell doubling time, the different time points in culture needs to be defined. Cell morphology to be examined using different microscopy techniques like fluorescence microscopy, confocal microscopy. These microscopes, let me tell you at this point, are different than widely used than the microscopes which we use in the material science discipline, because in the material science discipline we use mostly scanning electron microscope or transmission electron microscope, but for the cell biology related analysis one has to use the fluorescence microscope or confocal microscope, and also one has to do gene expression analysis. Based on the in vitro screening, which often involves genotoxicity analysis, genotoxicity means that is a DNA level toxicity, only few material compositions should be considered for the in vivo biocompatibility. Since all these in vitro in vivo things are interlinked, so we have to understand these things in a much more greater details. That is why I keep coming back to some of the discussion points again and again through this module discussion. Now, coming to the in vivo biocompatibility, as I said that selection of an appropriate animal model with respect to the final targeted application is the first key point then one has to address. Second one is to find out that what is the short term versus long term implantation, and whether what kind of defect model you are using, whether it is a critical size defect, whether it is a segmental defect, whether it is a femoral defect, all those things, and assessment of inflammation around implanted time, and also the tissue response. Now, tissue response here one has to do some different analysis, what is called histology. Histology means that is the scientific analysis of the structure and morphology of the different tissues in a thin tissue, in a thin histology slides, and this tissue response is important because there you can see that what an implanted material, whether it can cause toxicity to the cells in the neighboring tissue and so on. So, this point has been mentioned here, that is histological analysis of the tissue sections to investigate neobone formation or cellular activity at any synthetic implant and bone host bone interface needs to be conducted. Now, this point I have mentioned very briefly in an earlier slide, that is in vivo toxicity analysis. In vivo toxicity analysis means that heap simulator testing I have mentioned just few minutes ago. I will discuss it little bit more details later. Now, heap simulator testing always generates some finer wire debris particles. Finer wire debris particles essentially are generated because of the continuous friction and wear at the interface. Now, these wire debris particles, when they are transported to different tissue structure or different organs, they can cause toxicity of those vital organs, and this potential toxicity after biodistribution needs to be evaluated. And then one of the key things in the preclinical study is that it requires animal ethical committee approval. Without approval, one should not conduct this kind of animal testing. Okay. Now, to some of the other key points which has been discussed in this module and the previous module are as follows. The assessment of the biocompatibility properties should be a primary concern irrespective of the mechanical and physical properties of the material. Now, this point has been mentioned before also. So, what I have mentioned, suppose you have two different type of materials A and B. Suppose A has a very good, let us say, strength properties of 500 megapascal, B has a relatively less strength properties like 300 megapascal. This is your strength, typically we define in material science as sigma. Then there is the elastic stiffness, we typically define in the material science by E, and the elastic modulus, let us say A has a elastic modulus of around 210 gigapascal, and here E is a elastic modulus of 70 to 80 gigapascal, let us say 70 gigapascal. This is sigma is a 300 megapascal. Now, a material cause some inflammation, major inflammation or foreign body response and toxicity in vivo. I put a star. However, B material shows acceptable biocompatibility property both in vitro and in vivo. If you are a purely metallurgist or material scientist, then definitely you will choose the material A, not B, but that option is wrong as far as the biocompatibility or biomaterials is concerned. It does not matter whether the strength property is little bit inferior or elastic modulus is less. What is more important is that whether this material has a good biocompatibility both at the cell level and tissue level. So, with this very simple example and very hypothetical example, I hope I have been able to convince you that how this biocompatibility property should be given more priority. Now, one more point that needs little bit more discussion is on that elastic modulus. Now, elastic stiffness essentially means that the ratio of the stress to strain. Now, if the elastic modulus is, why it is more important, your material is always surrounded by host tissue or host bone structure, right. So, you have your implant material and surrounding this is your shaded region is the host bone structure. Now, if there is a large difference between the host tissue and in your synthetic material, then what would happen? If the implant material has a large elastic modulus, then implant will carry most of the weight or most of the load, whereas the host tissue will carry much less load under a given biomechanical stress situation. And that is not desirable, because that will lead to the implant loosening or aseptic loosening. So, there should not be large difference in terms of the host bone elastic modulus and implant elastic modulus, that is very key. So, 210 gigapascal, certainly the bone stiffness is much less than this 210 gigapascal. So, this is the typical stiffness of steel based materials like stainless steel or any steel for that matter has a elastic modulus of 210 gigapascal. So, two things I have mentioned here. One is that you have to give more importance to the concept of biocompatibility, that is point number 1 or priority number 1. Once a material is biocompatible, and if you have to select from a group of biocompatible materials like PQRS, and if among the PQRS, if so now if you have this 4 material PQRS and these all are biocompatible materials, then but certainly this PQRS depending on the different material composition, they have a different elastic stiffness value or elastic modulus value E1, E2, E3, E4, and your bone elastic modulus let us say EB. So, now you have to see that which one is very close to EB, and if for this hypothetical example, if the material R elastic modulus E3 and E3 is very close to that of the EB, that is the bone elastic modulus, then you have to choose the material R, and in material R I should tick, because it is expected for this kind of orthopedic applications or bone regeneration applications, which is essentially done for restoration and repair of any bone defects or any osteochondral defects, then you have to see that whether this material has elastic modulus or elastic stiffness which is extremely close to that of the human natural bone. So, I hope I have put some emphasis on what mechanical property one should look at and how this biocompatibility property should be given its due and central importance in selecting a material for biomedical applications. So, with this we will come back to the next module for more discussion or continuing discussion on tissue engineering and scaffold and so on and so forth. Thank you.